Welcome to this presentation on biomedical research design, where we frame reliable, ethical, and impactful scientific inquiry. In the next slides, we will explore how careful planning turns questions into credible evidence, why design matters for patient safety and scientific progress, and the practical steps researchers can take to improve reproducibility and real-world impact. This section introduces the foundations of biomedical research design, setting out the core principles that guide rigorous studies. Think of this as laying the groundwork, clear objectives, appropriate methods, and ethical safeguards that together ensure findings are trustworthy and useful for advancing medical knowledge and clinical practice. Research design is the strategic framework that maps a study from hypothesis to conclusion, guiding every decision from methods to reporting. A robust design makes results valid, reliable, and interpretable across contexts, so researchers can draw meaningful, reproducible conclusions rather than producing ambiguous or misleading outcomes. Good research design protects resources, preserves scientific integrity, and enables precise, measurable discovery. Poor planning wastes time and funding and can produce findings that mislead practice, while ethical and methodological rigor ensures participant safety, data quality, and results that truly inform healthcare decisions. The key elements of research design start with clearly defined objectives and testable hypotheses, then progress to appropriate data collection methods, thoughtful sampling strategies to ensure representativeness, and pre-planned analysis together with ethical safeguards. These components work together to make studies scientifically sound and ethically responsible. The research design process follows a clear progression from an initial question, through study design and defining the population, to data collection methods and analysis. Each stage builds on the previous one to ensure methodological rigor, minimize bias, and deliver reproducible outcomes that support confident conclusions and practical application. This section explores the different types of biomedical research studies, highlighting how each approach answers different questions. From laboratory-based experiments to human clinical trials and population-level investigations, the study type you choose shapes the methods, ethical considerations, and the kinds of conclusions you can draw. Biomedical research spans three main areas: basic research that uncovers cellular and molecular mechanisms in the lab, clinical research that tests interventions and measures patient outcomes, and epidemiological research that studies disease patterns and risk factors across populations to inform public health strategies. Experimental designs involve active manipulation to test cause and effect, with randomized controlled trials as the gold standard for causal inference, while observational designs involve careful measurement of naturally occurring phenomena across cohorts, case control studies, or cross-sectional snapshots to explore associations without intervention. Designing clinical trials requires rigorous control and randomization to minimize bias, clear definitions of patient populations such as intention to treat or per-protocol groups, and thoughtfully chosen primary and secondary endpoints that measure efficacy, safety, and meaningful patient outcomes throughout the study. Clinical trials progress through a series of phases that build on one another, starting with preclinical lab and animal studies, then Phase 1 safety testing in small groups, Phase 2 dosing and preliminary efficacy, Phase 3 large-scale efficacy trials, and finally Phase 4 post-market surveillance to monitor long-term outcomes and rare events. This section focuses on improving experimental design while upholding ethical standards, emphasizing collaborative planning, stakeholder engagement, and continuous refinement of methods to reduce harm and improve data quality so that research both advances knowledge and respects the rights and welfare of participants and animals. The three RUPs, replacement, reduction, and refinement, remain central to ethical animal research, encouraging alternatives where possible, minimizing the number of animals used through optimal design and statistical power, and refining procedures to reduce pain and distress while improving living conditions and humane endpoints. Common pitfalls in experimental design include failures in randomization or blinding that introduce bias, insufficient sample size and power that prevent detection of real effects, and overlooking important biological variables or confounders, all of which can limit generalizability and lead to misleading or unusable results. Several tools can enhance design quality, from free online experimental design assistants that guide sample size and randomization decisions, to reporting checklists like Arrive that promote transparent in vivo reporting, and pre-planned statistical support to prevent data dredging and ensure robust analysis methods are selected in advance. Reporting standards in animal research are often incomplete, with many studies failing to describe key elements such as randomization, blinding, power analysis, or sample size calculations, and this gap undermines reproducibility and highlights the urgent need for stronger adherence to established methodological reporting guidelines. Now we move to practical steps for designing your study, focusing on a clear, stepwise workflow that starts with defining focused research questions, selecting the most appropriate study type, developing standardized protocols, planning analysis and ethical approvals, and preparing for dissemination of results to stakeholders and the scientific community. A step-by-step workflow helps translate ideas into rigorous studies by first defining answerable research questions, then choosing a study design that matches objectives and resources, creating detailed protocols and quality controls, planning statistical analyses and ethical reviews, and finally preparing how findings will be shared and implemented. To illustrate these principles, consider a clinical trial case study where the objective is to assess efficacy and safety of a new therapy. The design is randomized, double-blind, placebo-controlled to reduce bias. The sample is defined with strict inclusion criteria to ensure comparability, and outcome measures include validated primary endpoints and relevant secondary safety metrics. In closing, rigorous biomedical research design builds a foundation of trust, protects ethical standards, and shapes future impact by informing clinical practice and patient outcomes. Embracing available tools, transparent reporting, and continuous improvement turns careful design into a moral and scientific imperative that advances human health.